balance sheet of aerobic respiration. So in aerobic respiration, how many ATPs are synthesized during oxidative phosphorylation or by substrate level phosphorylation? How many ATPs are synthesized? How many here NADH2s, FADH2s, how they are giving here ATPs? So let us discuss here. If we assume the glucose is the starting point in the aerobic respiration. So sometimes in the aerobic respiration, here different materials are also involving, like fats or proteins. So different substrates are also entering into the aerobic respiration. But here if we assume some assumption, based on some assumptions, here the aerobic respiration, here if the glucose is the starting point, the glucose is the starting material if it is involving in the aerobic respiration. And here the glucose is directly, it is completely oxidized into carbon dioxide and water. So it is complete oxidation. In the complete oxidation, when one glucose molecule is completely oxidized, how many ATPs are synthesized? So that is generally 36 ATPs. So glucose is when completely oxidized, here 36 ATPs are synthesized. How 36 ATPs? Where they are synthesized? So let us discuss here. Here based on some assumptions, here in the glucose oxidation, complete aerobic respiration or complete oxidation, here the starting material is glucose. Glucose is the starting point. In almost all aerobic respirations, here all carbohydrates are converting into glucose. So the glucose is the starting point or starting material in aerobic respirations. But sometimes other materials are also involving, like fats or proteins, organic acids. But here the other materials are entering into the aerobic respiration in different steps, not in the form of glucose. So the glucose is the starting point, and next one is here no materials are, no intermediates are involving in the synthesis of other materials, other compounds. No intermediate. In the glucose is converting into glucose 6-phosphate, fructose 6-phosphate, different components are produced in aerobic respiration. But the intermediate components like glucose 6-phosphate or fructose 6-phosphate or phosphoglyceraldehyde or dihydroxyacetone phosphate or pyruvic acid. So different components. So they are continuously involving in the aerobic respiration. So the no intermediates are involving in the synthesis of other material production. They are not withdrawn. So continuously the steps are taking place. Means glycolysis. After glycolysis, pyruvic acid is synthesized at the end of glycolysis. So it is undergoing oxidative decarboxylation. Next involving in the Krebs cycle. So in this process, carbon dioxide is released. So here different steps: glycolysis, pyruvic acid, oxidative decarboxylation, Krebs cycle. So after glycolysis, it is taking place. After this oxidative decarboxylation, Krebs cycle. So one by one, continuously it is taking place. Stepwise reactions are going on. No intermediate products are involving in the synthesis of other compounds. Next, other components are not entering into the aerobic respiration. No alternative components. No alternative components are entering the respiratory pathway. So the aerobic respiration, it is stepwise reaction continuously from glucose. Glucose is a starting material. From glucose to here, glucose is splitting into pyruvic acid, undergoing oxidative decarboxylation. Next, the Krebs cycle, continuously the oxidation reactions are taking place. Here, intermediate products, they are not involving in the synthesis of other components. The components are not withdrawn from the respiratory pathway. And no alternative components. Other components are not entering into the respiratory pathway. So, based on these assumptions, here we can calculate the balance sheet of aerobic respiration. How many ATPs are synthesized in aerobic respiration? Based on the previous assumptions, here ATPs, how they are produced in different steps. How many ATPs are produced? Here in the aerobic respiration, the first step, glycolysis. In glycolysis, by substrate level phosphorylation. So, what is substrate level phosphorylation? Here it is directly a direct level phosphorylation, or ATPs are synthesized directly. In glycolysis, total 4 ATPs are synthesized directly. So, the 4 ATPs are produced. So, between the 1,3-bisphosphoglyceric acid to 3-phosphoglyceric acid and phosphoenol pyruvic acid to pyruvic acid, at 2 points 4 ATPs are synthesized and 2 ATPs are consumed, 2 ATPs are utilized. So, where they are utilized? The first and third steps of glycolysis. From glucose to glucose 6-phosphate, 1 ATP is consumed and a fructose 6-phosphate to fructose 1,6-bisphosphate. There also 1 ATP is in glycolysis. 4 ATPs are produced by substrate level phosphorylation, 2 ATPs are consumed. So, the net gain of ATP is only 2. And in glycolysis, here NADH2s. NADH2s are produced only 2. From phosphoglyceraldehyde to 1,3-bisphosphoglyceric acid. Here 2 NADH2, at that point 2 NADH2s are produced. Here no FADH2s are produced in glycolysis. So, as we discussed in electron transport system and oxidative phosphorylation, glycolysis NADH2 is equal to 2 ATPs. So, 1 NADH2 is equal to 2 ATPs. 2 NADH2s are equal to 4 ATPs. So, in totally in glycolysis, here the net gain of ATP is 4 plus 2, 6 ATPs. The second step of aerobic respiration, pyruvate to acetyl coenzyme A. In pyruvic acid oxidative decarboxylation, here 2 NADH2s are produced. 2 NADH2s are produced. And each and every NADH2, it is these two reactions are taking place in the mitochondria. So, mitochondrial NADH2 is equal to 3 ATPs. And in this second step, no substrate level phosphorylation, no FADH2s are produced. In the second step also, the net gain of ATP is only 6 ATPs. Next, third step, Krebs cycle, also taking place in the mitochondrial matrix. Here, in Krebs cycle, by substrate level phosphorylation, here 2 ATPs are produced. The 2 ATPs are produced between the succinyl coenzyme A2 succinic acid. There 2 ATPs are produced by substrate level phosphorylation. GTPs are also equal to ATPs. Here 2 ATPs are produced. And in the Krebs cycle, by oxidative here NADH2s and FADH2s. Here NADH2s completely 6 NADH2s at 3 points. 6 NADH2s are produced. The 3 points are here isocitric acid to oxalosuccinic acid and alpha ketoglutaric acid to succinyl coenzyme A. Next malic acid to oxaloacetic acid. So here at 3 points, at every point 2 NADH2s. At 3 points 6 NADH2s. So each and every NADH2 is equal to 3 ATPs. Next FADH2s. FADH2s are produced between the succinic acid to fumaric acid. At that point 2 FADH2s are produced. So this FADH2 is equal to 1 FADH2 is equal to 2 ATPs. So total in Krebs cycle by substrate level phosphorylation 2 ATPs. Here when NADH2s, total NADH2s are 6. When they are undergoing oxidation or in the oxidative phosphorylation, so these are producing 18 ATPs. So each NADH2 is equal to 3 ATPs, total 18. And FADH, 2 FADH2s, each FADH2 is equal to here 2 ATPs. So total here 2 plus 18, 20 plus 4, 24 ATPs. In the total aerobic respiration, the total net gain of ATPs 6 plus 6, 12 plus 24, 36 ATPs. That is the net gain of ATPs in aerobic respiration. Here by substrate level phosphorylation 4 ATPs are produced. 4 ATPs. And by oxidative phosphorylation, when these are involving in the oxidative phosphorylation, here 4 plus 6, 10 plus 18, 28 and here 4 ATPs. 4 plus 28, 32 plus 4, 36 ATPs. So here it is the net gain of 36 ATPs or net gain of ATPs in aerobic respiration. If one glucose is involving in the aerobic respiration up to here one glucose is oxidized completely. When one glucose is oxidized completely, that is giving 36 ATPs. So based on some assumptions, here if the starting material is glucose, when it is completely oxidized, it is giving 36 ATPs. So it is about a balance sheet of aerobic respiration. One glucose is equal to 36 ATPs.